Okay, we are back talking about polypeptides put together by what kind of a reaction? A dehydration reaction. So you've got your amino acid, another amino acid, you're going to lose some water here, and you're going to make a polypeptide. Now, when these cells link amino acids together by a what? Dehydration reaction, forming a peptide bond. So the bond is actually this bond right here that gets formed. And this creates a long chain called a more than one, peptide. So it's a peptide bond forms a polypeptide. And that's all through the magic of a dehydration reaction. Now, these are very specifically folded. A functional protein is one or more polypeptide chains precisely twisted and folded and coiled into a very unique shape. If it doesn't fold correctly, it causes disease.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
We'll talk about the 4 levels of structure in a minute. So they are joined together by a dehydration reaction that forms this peptide bond. How is it possible to make a huge variety of proteins from just 20 kinds of amino acids? It's kind of like the English language. We have 26 letters, and it makes up an infinite number of words. So that is how your 20 amino acids make up the variety that is you, essentially. Almost everything in your body is made with protein from amino acids.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Okay, this is going to make sense. Wait for it, because we're going to talk about disease. Okay, so the primary structure is the amino acid sequence of each polypeptide determines its 3-dimensional structure. So the sequence, the letters, are eventually going to make a structure because the functional groups that come off those are what's going to bond with each other. The protein's 3-dimensional structure enables the molecule to carry out a very specific function. Do you see what I'm doing there again with structure and function? Nearly all proteins work by recognizing and binding to some other molecule. So here you have your primary structure, and you're going to see how all of these actually create bonds that will allow you to see its later structure. So one amino acid, let's say this is alanine, is your structure. And that alanine very specifically bonds to something else that creates a structure. A slight change in the amino acid. So if we go back here and change this alanine to something else, a slight change in the amino acid sequence affects a protein's ability completely to function because it will not fold right. The substitution of one amino acid for another at a particular position in a hemoglobin molecule. So we're substituting one amino acid. Really, we're substituting one gene that alters the function of the amino acid. So we have one of these whole amino acids. Look how many there are. There's a bunch of these, and we're just messing with one of them. That one in hemoglobin, which carries oxygen, causes sickle cell disease. It's an inherited blood disease. This is what it looks like. So normal hemoglobin, we have the alanine that's here. It makes a normal peptide, which makes a normal red blood cell. We have one wrong amino acid. It doesn't fold correctly. This string of beads does not fold correctly, and that makes your red blood cell what's called sickled. It's like this, and so this hooks on things, which causes you to be very ill. So that is only one amino acid change. That's why we care about amino acids. I know you're like, why do I even care about amino acids? That's why, because it causes diseases. One difference in structure is enough to cause the protein to fold into an entirely different shape, which alters its function, which in turn causes diseases. Misfolded proteins are associated with many diseases, including severe nervous system disorders. There's one in cows that cause, and it leads to brain damage. A protein shape is sensitive to the environment. An unfavorable change in temperature, pH, or other factors can cause protein damage. So when you have a high fever, especially above 103, your proteins start to denature. They start to unravel, and it can cause permanent brain damage because the temperature change is so high. Proteins are made by nucleic acids. Nucleic acids are macromolecules that store information. They provide instructions for building proteins. There are two types. These should not be brand new words for you. DNA, RNA. Deoxyribonucleic acid, ribonucleic acid. So those are the two types of nucleic acids. Nucleic acids will ultimately make protein, the genetic material that humans and other organisms inherit from their parents. Basically, it goes DNA, folds into a chromosome. Actually, it folds around these histones, folds into a chromosome. Those chromosomes are in the nucleus. So DNA folds around a histone, makes a chromosome. The chromosomes are in the nucleus is in the cell. So the DNA resides in the cell as one or more very long fibers called chromosomes. So here we have a gene. This is a chunk of DNA that's right here in your DNA, and that's going to get folded around and put into your chromosome, right? A gene is a unit of inheritance encoded in a specific section of DNA that programs the amino acid sequence of a polypeptide. Nucleic acids. Those program instructions are written in a chemical code that must be translated from nucleic acid language to protein language. How does that happen? Whoops. The DNA or the RNA ultimately form amino acids, which form a protein. So your DNA or your RNA, whichever nucleic acid you're working with, will form an amino acid, and that will form a protein. So do you see now how sickle cell is inherited through the DNA? The wrong nucleotide here creates the wrong folding in the amino acid sequence, which creates the wrong protein and gives you sickle cell. Nucleic acids are polymers made from monomers called nucleotides. Each nucleotide has three parts, right? The sugar, deoxyribose or ribose, a thymine or a uracil or a guanine, and a phosphate group. So basically it has a phosphate, a sugar, and a base. It looks like this. Basically it's A, C, T, A, G, T, C are your types for DNA. For RNA, there'll be a uracil there. And essentially it looks like this: an adenine, a guanine, a thymine, and a cytosine. Dehydration reactions, oh my gosh, it's like I'm saying the same thing over again. Link nucleotide monomers into long chains called polynucleotides. They form covalent bonds between the sugar of one nucleotide and the other, and they form a sugar-phosphate backbone. Bases A, T, C, and G hang off the backbone like appendages. It looks like this: your A, your T, your C, your G. It forms a double helix. Bases along the DNA strand, and you get A pairs with T, G pairs with C. The structure looks like this. There are many similarities. Both are polymers of nucleic acids, and both are made of nucleotides consisting of a sugar, a phosphate, and a base. There are three important differences between DNA and RNA. This is definitely going to be a test, so write those down. A ribose, a uracil, and it's single-stranded. Those are the three.